I have the pleasure of introducing Dr. Julie Johnson this afternoon. She's the Assistant Vice President for Research Compliance and the Biosafety Officer for the Biosecurity Research Institute since 2006, and she serves as the responsible official for select agent use on campus. Prior to working at K-State, she was the Campus Biosafety Officer at Iowa State University for 11 years. Julie has a bachelor's degree in chemistry from St. Olaf College and a master's degree in biochemistry and doctorate in molecular, cellular, and developmental biology from Iowa State University. She's been a certified biosafety professional since 2001 and an active member of ABSA International for 29 years, previously serving as a council member and on the Select Agent Users Task Force, and currently serving on the CBSP Exam Development Board. She also led the non-federal portion of the team that wrote the Agricultural Pathogen Biosafety Appendix for the BMBL 6 edition.
Good afternoon, everybody. Thank you for coming. I know IBC protocols are not the most exciting topic in the world, but they're important things. So hopefully I can convince you why they're important. And for those of you who this might apply to directly, some tips on how to make this. And we don't have a laser pointer, so I'm staying here in spacebar. Okay. So the compliance committees are run out of the Research Integrity, Compliance, and Security, or ORICS. So that is, for those of you who have been around a while, that is a fairly new name for what you used to know as ERCO, or University Research Compliance Office. Part of the reason for that name change is recently that has expanded to include not only the compliance committees, but also research integrity and research security. And that the main office is located in Fairchild Hall on the halfway up the stairs mezzanine. So to give you an idea of what makes up that office, so Brad Woods is our Associate Vice President for Research. He leads the ORICS office. Heath Ritter is the Compliance Director. And then we have administrators for the three main compliance committees. The IRB, which McKenna DeHart manages, is Institutional Review Board, which if you're not familiar, that governs the use of human subjects. So all approvals for that. Anna Lemon manages the Animal Care and Use Committee, the compliance for animal research-related compliance. Natalia Costabal, who I am filling in for today, she's out on maternity leave, is the IBC program manager. So we're going to focus on the IBC today. Institutional Biosafety Committee, which we'll talk about in detail. And then export controls is managed by Lisa Brummett. And the biosafety team at the Biosecurity Research Institute is actually part of ORICS, although we are pretty much 100% dedicated to Biosecurity Research Institute, except for a little bit of. So ORICS oversees human subjects research, animal research, and I'm going to skip the middle one, export control program, and dual use research of concern. Dual use research of concern is kind of a subset of what IBC oversees. So the IBC legally, officially oversees research using recombinant or synthetic nucleic acid molecules, toxins, or infectious agents. Dual-use agents are a specifically regulated list of infectious agents that are human pathogens, where there's more concern of used for, I guess, negative means. So that's something that the IBC kind of does that initial screening of that. And if that requires further oversight, which currently we don't have that issue on campus, then there is a committee for the dual use research of concern that does follow up for that. Okay, so if you are a scientist, you may sometimes think that having to get approval from compliance committees for all the things you do is tedious and like, why do I have to do this? It's not just because we want to like employ people to fill up an ORIX office or micromanage scientists. It is because there are lots of federal regulations that require oversight of research. And a big part of what that means is if we aren't compliant with those federal guidelines, we do not get to keep grant money. And for this, specifically, IBCs originated by the NIH guidelines for research involving recombinant or synthetic nucleic acid molecules, which is overseen by NIH, that requires that any institute receiving NIH money that does any of this kind of work must have an IBC. And that doesn't mean only if you have NIH funding. It's like if this institution has any NIH funding, the entire institution has to be in compliance with this. And we do not want to lose our NIH funding or any other federal funding. Most federal grants are going to have some clause about being in compliance with all federal regulations in order for us. So if nothing else, if not for safety or any other reason, we can't do research without being compliant. So the goal of ORIX is to help everybody ensure that we are compliant and keep that funding going and keep. So we're required to establish an IBC. And those guidelines describe who has to be on that committee. So that includes specific expertise in assessment of risk to the environment and public health, expertise in biocontainment, and of course, knowledge of institutional policies, applicable laws and standards. So we have to, figure out who's on the committee from the requirements. So at Kansas State University, the IBC is appointed by the Vice President for Research. Here in addition to only things that have to do with recombinant or synthetic nucleic acids, we also review anything involving microbiological agents. That includes things affecting humans, animals, or plants. Not just humans, we are an ag university, so we care a lot also about things like wheat and pigs and cattle. And this is really common among most IBCs. Also do oversee that because there are different regulations that might not require a committee, but they require that your institution is compliant with safety practices regarding biological materials. And that really fits well in with the IBC. Some kind of subsets of what that oversight means is there's a list of select agents and toxins, and that's what I'm the responsible official for here on campus. So that's a separate federal regulation of things that are more highly regulated because they could be thought to be potentially used for criminal or terrorist use. So we regulate those a lot more. Most of those things also require high containment, so they're all used at the Biosecurity Research Institute on campus. And then anything that would fall under the potential dual use implications, and there's quite a bit of overlap between that select agents list and the dual use. The dual use focuses on human illness, things that could cause human illness primarily. And then some things that sort of fit in there, human blood, tissues, and cell lines. Because of bloodborne pathogen regulations, we have to have appropriate safety protocols and training in place for those. And then tumorigenic cell lines or cell lines that might contain human pathogens that have been used to immortalize them are also covered because there are some safety practices that kind of fit related to that. So the IBC also does laboratory inspections for all laboratories on campus that operate at BSL-2 or ABSL-2. They do also come out. So the only things that are different than BSL-2 or ABSL-2 are the BRI facilities, which are BSL-3, ABSL-3s, ABSL-3 Ag. They do also come along when we do my team does that internal inspection. So It's just, that's more of a joint. Okay. So let me ask, how many people here have ever filled out an IBC? Okay, so you're a little bit familiar with the IBC. So first thing you're going to do if you need to have IBC approval for work that you're going to do is go to the ORIC's website, go to the IBC section, and you can find all the information you need about that committee, how to submit an application, who's on the committee, all the different policies and things like that. So this slide kind of looks like this is only about human subjects research, but the software that is currently used for managing IBC protocols is called IRB Manager. And IRB stands for Institutional Review Board, which is the committee that oversees human subjects research. So this software is originally designed for that, but it's used here for animal care and use and biosafety and human subjects protocols. So you're going to go to that IRB Manager website, and that's where you can log into IRB Manager to do that application. And if you have done the paper or PDF versions of application forms in the past, this really makes it easier because you can go back and change things more easily and then go back and look at all of your protocols much more easily. So when you do, we're going to go through that application form a little bit. And I'm going to point out some things where we often get questions. But essentially, you're going to go to this login that's on that web page. Note that there is also, can I point it with the mouse? Okay, so this link that's also on the web page is the Investigator's Guide to the IRB Manager. That's essentially the user manual, and it's very helpful. I use that as an IBC member. There's also one that's for from the management side of it. I use that manual when I'm like, how do I get to this information? So it is very useful and very straightforward. So just a note that that's there. So essentially, what happens after you submit an application is things that are only required by Biosafety Level 1 that are exempt from NIH guidelines. Those get just reviewed by the IBC chair. They don't go through that whole committee process. Say about that. Oh, people ask us, why do we even have to have those reviewed? Essentially, what the IBC is doing with that is confirming that they really are exempt, because if you've read the NIH guidelines, which many of you have in having to get these kind of approvals, they're not super straightforward, and it can be confusing to determine, is it exempt or not. So in that way, the committee is essentially just confirming that it really is exempt, and then that's all that has to happen. If it's something that's non-exempt or anything that requires Biosafety Level 2 or greater containment, those then get sent to the full committee. The committee meets once a month, and we generally review between 4 and 12 protocols, depending on the month. If it's right before grant submission season, we get a lot more. So sometimes it's a lot more. And then the committee reviews that. They'll send you back comments. And then essentially Natalia will work with you to get those comments answered so you can get your protocols approved. Okay, so this is what if you haven't seen it, this is what that initial page looks like when you log into that IRB Manager. And you can look up, like everything under but you're going to start out with this create a protocol. So once you find a protocol, or once you get to this page, you're going to choose what kind. and then that you'll get, oops, that's on there twice, to the initiation of the protocol submission. So I'm not going to go through this whole protocol form with you, but Natalia picked a few areas that people usually sometimes get their protocols sent back to them or often have questions about. So we can, if you know this stuff ahead of time, then you know, you don't want to get your protocol sent back to you for revision and not reviewed, you know, for an extra week or two because you misunderstood a simple question. If you can, you know, get that answered ahead of time, that, you know, helps you get it approved more quickly. So we just kind of went in order with this. So if you're going to have more than just you doing research, which of course you are, you have graduate students, postdocs, lab technicians, you need to list all of the participants in the who will be on the research study. The reason you have to list everybody is there are regulatory requirements for required training. And there may be requirements for example, for vaccinations or some kind of medical approvals, things like that, depending on what you're working with. So you need to add them. A tip to know is that there's this drop-down box here. So if their name doesn't pop up there when you look for it, you need to have them log into the system one time. Once they're logged in, then the rest of the time if you're here for 30 more years, their names will always be in there. But initially that has to happen. Don't just leave them off because that will delay things. There's a question that asks you about where you got your biohazardous agents. This one, we often have people answering it incorrectly. So the first two parts are, you know, do you possess it or not? If you don't possess it, where are you going to get it from? But the questions about permits often get answered incorrectly. So are transport permits required for any of these agents? If you don't know, just call us up and ask. There is a whole section on the IBC web page that talks about permit requirements. If you still don't understand after that, just call the office and we will help you figure that out. For example, the different kinds of permits you might need are CDC requires import permits if you're getting human pathogens from outside the country, and for a short list of pathogens, even if you're subsequently transporting them within the country. And sometimes going through that takes experienced helper to help you figure out what the answer is. And then what we see most of is things that require USDA permits, and that can be for either livestock and poultry pathogens or for different crop pathogens. And there's two different divisions within USDA that oversee those. And there is a sort of flow chart on the USDA APHIS website where you can figure that out, but sometimes that's questionable. One thing I've seen people get really confused about is that does require a permit is things like fetal calf serum. If you're getting it from a country where FMD is endemic, you have to have a permit because it needs to be screened to make sure it doesn't have FMD. Even if you know that it doesn't have FMD, if it came from country X, you may need a permit. So we can help you figure that stuff out. One other thing about transportation permits, if you already possess the material, but you had to have a permit to get it, you still want to answer that yes and include a copy of your permit to show that you did get the appropriate permit to receive it, because you're not going to have it shipped to you, but you did at some point. And then possession permits. Possession permits are rarely required. Most permits are only required to transport things because they're, for example, APHIS permits for livestock pathogens. They're required for transport, but once it's in your state, then it's your responsibility to make sure you're containing it properly according to your permit instruction. But they don't get additional permits if you give it to the person down the hall who's also doing research, things like that. And possession permits are really only sometimes required for USDA regulated plant pathogen, and sometimes they require you to have a permit as long as you possess the material, so you have to renew your permits even if you're not going to ship or receive anymore. Okay, so if in doubt, call us about permits. Okay, if you're going to be using animals, one thing that people also often get confused about is asking you, are you going to use more than one animal species? So you may be studying an infectious agent in mice and in pigs, or also cattle. For every animal species that you do, you have to complete a vertebrate animal appendix, which is a separate form that's linked. So if you're using multiple types of animals, you need to do a separate appendix for each of those. Much easier that way. Don't try to put something that requires an ABSL3 egg room and something that requires just an ABSL3 small animal vivarium on the same. It'll be super hard to answer the questions with multiple things. And we have to look at it because really a lot of that is going to the CMG, which is our centralized animal care, for their evaluation of how they need to have their staff work safely in those rooms. So this page is about dual use, is the dual use question page. But this is really showing us a couple of tricks for when you're doing these forms. So there's a lot of questions on the IBC. You may fill out part of it and then wait till the next day to do some more. And you may want to have your lab manager fill out some sections, or somebody else in your lab. You can do that. And all you need to do is go into the form and use this share form access button here, and that will pop up this window, so you can email it to whoever you want to also be able to do edit. Very helpful. So also, this box is at the top of every page. So if you want to jump back, if you think, oh, I forgot to put something in the permitting section, you can jump back to that. Or if you want to skip a section, fill out others, come back, you can skip between sections here. The one thing that you can go back to sections. It will not let you, so you hit next to go to the upcoming section. It will not let you hit next if you've left anything blank. So if you want to expand later, you should put at least something in there. And so it doesn't think you skipped a question, but you have to answer any question. If it's not applicable to you, put NA in there. Otherwise, it won't let you go forward. So that's tips and tricks for filling up the applications. So the other part of what the IBC does is it has to do lab inspections to follow up and make sure that people really are with agents the way that they say they are in their applications. So if you are filling out a new IBC application and the site where you're doing work has never been inspected before, you're going to first have a lab inspection to make sure that you have the appropriate facilities. And then we do follow-up confirmation lab inspections as needed. And we try to do those once a year. Try to do all the labs at least once a year as a follow-up confirmation lab inspection. So things that you can do to help yourself with this lab inspection this lab inspection program is part of the IBC website. So you can download the different checklists that are used for the inspections here. So you can kind of pre-inspect your own lab. And this is something I've been telling this to people for years. It's like lab inspection checklists are not only for making sure that an inspector doesn't find something in your lab. Lab inspection checklists are valuable for you to use to say like, hey, have I done everything I need for all of us to be working safely in my lab? It's not yes, we have compliance people that are coming around and confirming that you're doing that. But our purpose for doing it is for our own benefit, not compliance people. As scientists, your purpose for following a lab inspection checklist is to kind of remember that you remember all the things that you should be doing to keep yourself safe and your coworkers safe. It's not so you can make sure you don't get in trouble. Because it's not about getting people in trouble. It's about making sure you haven't forgotten something. Because you know how we work with something every day and we kind of forget how hazardous it is. So it's kind of more like a self-reminder. But the inspectors will look at this. So these checklists are there for your use, whether or not you have an inspection coming up. And we have different ones for different containment levels. And at the bottom of the slide, you'll see, you don't see the whole link, but there's a link for an actual video about kind of what to expect during an inspection. Yeah. And this is just an example of that checklist. So some of the things inspectors will also look at is that you have appropriate signage on your lab doors. This standardized signage is, can you get through the HNS website? So having a good standardized signage like this on all the labs helps people consistently know what the hazards are. So if everyone who works in a lab building is familiar with that, you know where to look to find out what the hazards are and what that all means. So that's some of the things that the IBC will look at as well. And I mean, just a note that some of the things, everything isn't only biosafety, it's just safety in general, including things like, are you checking the eye washes? Are you having security in your lab? Things like that. That's not necessarily infectious agents or recombinant DNA specific. Another important thing that you will do as the first time you're ever having something that requires IBC approval is you're going to develop a lab biosafety manual. That is something that will be looked at during an inspection, but this is something that there's a template on the IBC website where you can go in and you don't have to think, gee, what should I put in my biosafety manual? It has, you know, all the checklists for you to fill that in of what's in there. And then when we come to your lab, we'll expect it to look just like this, which doesn't look like they do work in there, but I thought that was a great, I don't know where Natalia found that, but it's a great picture of a super clean lab where no activity is done. And this is the link to it, but if you go on the IBC website, you will find that. Super long. But so that gives you just a template to just fill in. Okay, so just a reminder so ORIX is located in Fairchild, and if you have questions about any of the compliance committees, because there's a good chance you're doing stuff that requires oversight or assistance from not just the IBC, but other compliance committees, all you have to know is comply@ksu.edu. That goes to the ORIX office and will get forwarded to the appropriate people who can help you. And I guess I would just say overall, don't think of ORIX as only an oversight or a hurdle to jump through so you can do your research. Think of it as somebody that can help you to get through all the red tape you have to do to be safe and your grants. Be compliant. So what questions do you guys
get a lot of. We share information.
Point. Try to minimize duplication of effort on campus.
Different things.
Yes, but they're looking at different things. So, IBC, yes, you're gonna get an inspection from APHIS if you don't already have a permit. They're gonna look at all the spaces. And yeah, IBC has to look at them for all the biosafety things. Often with animal things, IBC and ICAC try to, no, they don't. No. But if possible, do that, but it's not always possible. But ICAC has to evaluate for animal welfare things, which are, related but separate.
But this would only be regarding the transport.
I mean, what do you mean by
For APHIS, so.
Right.
And that covers
the APHIS permit?
the
Yes. Oh, you mean APHIS would request to see that?
Yeah.
No, but they still indications that the location is appropriate and that it has appropriate containment and practices. So I see what you're saying. So it's a core facility, but they would look at how they still have to look at that facility and how it's managed because you're going to be doing that work. And even if it's not under the PIs, but you are so as the PI with that permit, once you receive that material, you are responsible for that, for making sure it's in your lab, and you're responsible for making sure that it's used appropriately. If you're infecting animals at LARC, yes, LARC provides the animal care, but the research you're doing is done by your team, right? So you're still responsible for that safe behavior. And I mean, that's a little bit of a joint, you're not, but like if you were to observe that animal care personnel are practicing biocontainment, oh, well, that's did that answer your question?
This is an extra.
Yeah.
Shared spaces or inside.
How we handle share.
Or like work.
Oh, okay. So I wasn't sure what you meant by for service. So things like the diagnostic lab, right? Like they don't know what they're going to get. I mean, sometimes they get select agents and there's so as far as like IBC and diagnostic lab doesn't have IACUC, but as far as like IBC goes, for example, the diagnostic lab does have sort of like a generic IBC protocol that's kind of for unknowns and it's pretty broad and generic. And then, for example, they have special protocols. So say they get something in that they suspect is a select agent. They have, small number of specially trained people who will handle that case. And they essentially they don't have BSL-3 facilities, but they do BSL-3 practices in the BSL-2 facility with everything in a biosafety cabinet. They have to keep that material secure and everything and notify they notify me, they notify it's usually a veterinarian sends in a sample. Common in Kansas, cat or rabbit, tularemia. So, and then they have to ensure that material is properly destroyed. So they still are, kind of regulated, but a little, because you can't apply for a permit and IBC approval for like everything in the world. And the way the like APHIS permitting works, they also have a permit that's kind of general diagnostic things.
No longer. Facilities. Like I have a
Oh, okay.
core, I have this.
Okay.
No one else has a sample I could get. Those kind of. What kind of?
Okay. So like diagnostic labs and core facilities a little different in that
You know what's going.
Yeah, say, yeah, there's a core and there could be everybody in a department can use that core. That core, I mean, as far as a permit goes, if the material is going to be used in that core, even only sometimes and only by one person, that core would still get in. They're not going to, I mean, for permitting, they don't inspect like all the users. They're going to inspect the locations and how it's used. Calls. I guess
List for agent effect. Everything will come out. What?
Oh, so say somebody's going to take something out of the BRI.
It'll be inactivated.
That's inactivated.
It's
not going to come out unless it's inactivated.
Yeah.
But they're going to use it in a core facility.
Yeah.
It's not a select agent anymore. It's inactivated.
Right.
So it's or a common. It's like formalin fixation is sort of different. It's like it's the same. Like you might have pig tissues infected with African fever, but they've done a validated, verified inactivation by formalin fixing it. It's not an infectious agent in any way at all anymore. Your risk is the formalin. But a common thing we have is people will extract nucleic acids. And as long as they do a really rigorous process to ensure that there is no viable agent left in the extract before they can take it out of that building. So those then also, it would be, if I'm extracting DNA from Rift Valley fever, animal tissue infected with Rift Valley fever virus, that's DNA that is not infectious. And for example, you can't take out DNA from or nucleic acid for something that if the nucleic acid itself could be infectious in any way. So it could be segments of DNA. So if you had to list material, be a BSL-1 material. Yeah, if you're saying what?
You just need the letter.
Right. And so for things that are a select agent coming out of the BRI, we're legally required that they come with a certificate signed by the principal investigator. So that is something that the research team can keep and show, like, see, this things that aren't select agents, there is not a certificate requirement, but it still goes through the BRI biosafety team that we have to confirm that they used an approved protocol. They still have a validated protocol, not to the level of validation and re-verification do it. Other things that would, require bios. For example, Japanese encephalitis virus. It's not a select agent, but it still goes through, there's not that regulatory, but it's still end up. So you could have, I mean, the certificate is a really easy, like, certificate shows I did this file of documentation, but they could show the documentation that an email from BRI Biosafety saying, yes, if you've done this, this, and this, you can take it out of the building, or they could show this was activated using this